Dear students, welcome to the second lecture on carbohydrates. In this lecture, we will learn about the classification of the carbohydrates. I am Dr. Muzammil, Assistant Professor, Department of Biochemistry, Government College for Women, MA Road, Srinagar. In the previous video, I presented to you the definition of the carbohydrates. Now, in this lecture, we will see different classes of the carbohydrates. Students, remember that carbohydrates are classified mainly into four different groups. 1. Monosaccharides, 2. Disaccharides, 3. Oligosaccharides, and the fourth class is the polysaccharide. So first of all, let us see the monosaccharides. Monosaccharides are those carbohydrates which cannot be hydrolyzed any further. Means they are the simplest type of the carbohydrates. That's why these monosaccharides are also called as the simple sugars. The common example of the monosaccharides include glucose, fructose, ribose, and galactose. Now, these monosaccharides are further classified on the basis of one, the functional group present in them, and second, on the basis of the length of the carbon chain. First, classification on the basis of the carbonyl group. Monosaccharides, those monosaccharides which possess an aldehyde group are referred to as aldoses, and those monosaccharides which possess a ketone as the functional group are referred to as ketoses. So, on the basis of this, we'll have two types of the monosaccharides: aldoses having aldehyde as the functional group, and ketoses having ketone as the functional group. The second classification of the monosaccharides is on the basis of the carbon chain length. So the simplest of the monosaccharides are those monosaccharide which has a 3 carbon chain. The monosaccharide having a 3 carbon chain are referred to as trioses. Those having 4 carbon atoms are referred to as tetroses. Those possessing 5 carbon atoms are referred to as pentoses. Those carbohydrates having 6 carbon atoms are referred to as hexoses, and those having 7 carbon atoms are referred to as the heptoses. Now, students, look at these two different molecules. We have on your right a molecule which possesses an aldehyde group. So this is an aldose. On the right hand side, on the left hand side of your screen is another molecule which is again having 6 carbon atoms, but it has a ketone as the functional group, so it's an ketose. Here in this slide, I have given you examples of the monosaccharides having different carbon chain length. So the simplest of the monosaccharide is the one having 3 carbon atoms. So that's referred to as the triose. Example of the aldotriose is glyceraldehyde, while as the example of the ketotriose is dihydroxyacetone. Similarly, the example of the monosaccharide having 4 carbon atoms referred to as the tetrose is erythrose as aldotetrose, while as erythrulose is a ketotetrose. The monosaccharide having 5 carbon atoms referred to as the pentose. The examples under the aldoses include ribose and xylose, and ketopentose includes ribulose and xylulose. Monosaccharides having the 6 carbon atom referred to as the hexose. Example under aldoses include glucose, galactose, and mannose, while as ketohexose includes the fructose. Now, students, this is the structure of the simplest monosaccharide, that's the glyceraldehyde. Glyceraldehyde is taken as a standard molecule, and based on the position of the penultimate position of the hydroxyl group at the penultimate carbon atom, this glyceraldehyde is divided either as classified either as diglyceraldehyde or L-glyceraldehyde. Now let's look at the other molecules. This assignment of the D and the L to the different monosaccharides is determined on the basis of the position or the orientation of the hydroxyl group at the penultimate carbon atom. Remember that we number the molecules in chemistry starting from the functional group. So here the penultimate carbon atom is this one. You will see that this molecule has the hydroxyl group on the left side, so it will be referred to as the L sugar. While as this molecule, it has the hydroxyl group at the penultimate carbon present on the right hand side, so it's referred to as the D form, D sugar. So this D and the L is actually in reference to the glyceraldehyde. In D-glyceraldehyde, we have the hydroxyl group on the right hand side, and in the L-glyceraldehyde, we have the hydroxyl group on the left hand side. So the D sugar is the one which has the hydroxyl group at the penultimate carbon atom on the same side as is present on the D-glyceraldehyde. Similarly, the L sugar has the hydroxyl group present at the penultimate carbon atom on the same side as is present in the L-glyceraldehyde. Now, remember students, these monosaccharides also exhibit a type of an isomerism which is referred to as epimerism. Epimerism is referred to those isomers which differ from each other with regard to the hydroxyl group on a single asymmetric carbon atom. We have an example here. We have D-glucose and we have D-galactose. See, the positioning of the hydroxyl group at this asymmetric carbon atom in D-glucose is on the right hand side, while as the position of the hydroxyl group in the D-galactose is on the left hand side. So these two molecules, that's D-glucose and D-galactose, they differ in the orientation at just one asymmetric carbon atom. So if this is the case, then these two molecules are referred to as epimers. Okay, so D-glucose and D-galactose are C4 epimers. C4 because they differ in the orientation of the hydroxyl group at the fourth carbon. Okay, then these monosaccharides also exhibit another important feature that is known as optical isomerism. We know optical activity is a characteristic feature of the compound which possesses an asymmetric carbon atom. Just as we saw in the previous slide that the glucose is possessing the asymmetric carbon atoms. When a beam of a plane polarized light is passed through the solution of the sugars, these sugars rotate this plane polarized light either on the right hand side or to the left hand side. Okay, if this sugar rotates the plane polarized light to the right, then it will be referred to as dextrorotatory and will be designated as by the small letter D or plus sign. But if the solution rotates this plane polarized light on the left hand side, then it will be referred to as levorotatory and will be denoted by small letter D or minus sign. A racemic mixture contains an equal proportion of or equal concentration of D and L forms of the sugar molecules. So the racemic mixture does not exhibit any kind of the optical activity. So this was all about the monosaccharides. The second class of the carbohydrates are the disaccharides. Disaccharides, as the name suggests, are the are the carbohydrates which possess or are composed of two monosaccharide units. So here in disaccharides, the monosaccharides are linked together or joined together and form a disaccharide. Examples of the disaccharides include the sucrose, which is made up of glucose and fructose; maltose, which is composed of the glucose and glucose; lactose, which is composed of glucose and a molecule of galactose. Now, if we look at the structure of the first of these disaccharide molecules, that's the sucrose. Sucrose, it's a disaccharide, uh, okay, and it's composed of a molecule of the glucose and a molecule of the fructose. Remember, students, that the bond that is formed, or the bond that joins these two monosaccharide units together, is referred to as the glycosidic bond. Here in this case, that is sucrose, the bond is formed between the carbon number 1 of the glucose and the carbon number 2 of the fructose molecule. The orientation of the hydroxyl group at the the carbon number 1 in the glucose is alpha orientation, while as the orientation of the hydroxyl group at the carbon number 2 in the fructose is beta. So the bond is formed between the alpha form of the glucose and the beta form of the fructose. So it will be referred to as the alpha beta 1 to 2 glycosidic bond. Okay, sucrose, we know it's also known as the table sugar. Okay, and remember that sucrose is not a reducing sugar, means it does not possess a free functional group, because both the functional groups are involved in the formation of the glycosidic bond. The second type example of the disaccharides is the maltose. Maltose is a disaccharide which is formed by joining of the two glucose monosaccharide units. The bond that's formed is between the carbon number 1 of one glucose unit and the carbon number 4 of the another glucose unit. The bond formed, both these glucose molecules are in their alpha anomeric form, so the bond formed will be alpha 1 to 4 glycosidic bond. Remember that this maltose is produced from the breakdown of the polysaccharide starch and the glycogen. Maltose is present in high concentration in the cane sugar, so it's also referred to as the malt sugar. The third example of the disaccharides include the lactose. Lactose is a disaccharide which is formed by joining of a glucose molecule and a molecule of the galactose. Here in this case, the bond is formed between the carbon number 1 of the galactose and the carbon number 4 of the glucose. These two monosaccharides are in their beta anomeric form, so the bond formed is between beta 1 to 4 glycosidic bond. Lactose is known as the milk sugar because it's present in the milk. Okay, and we know that there's a disease associated if there's a deficiency of the enzyme which breaks this glycosidic bond, and that's known as the lactose intolerance. So that was all about the disaccharides. Now we'll move over to the oligosaccharides, which is the third class of the carbohydrates. Oligosaccharides. Remember, students, that oligosaccharides are formed when 3 to 10 monosaccharide units join together. If just two are joined together, they will be referred to as disaccharides. But if three are joined together, then it will be an oligosaccharide. even up to the 10 monosaccharide units. Okay, these oligosaccharides, they are an important part of the glycoproteins. Those proteins, glycoproteins, are the proteins which contain a protein part and a carbohydrate part. In most of these glycoproteins or glycolipids, the carbohydrate part is basically an oligosaccharide. Then we have the fourth and the last class of the carbohydrates. Those are polysaccharides. Polysaccharides are those carbohydrates in which there may be more than the 10 monosaccharide units joined together, and the number of the monosaccharides may go even up to thousands. Okay, these polysaccharides are further classified as the homopolysaccharides and heteropolysaccharides. The homopolysaccharides are those polysaccharides in which same type of the monosaccharide is joined together. Uh, Example include the starch, glycogen, and the cellulose. Then we have the second type of the polysaccharide. Those are referred to as the heteropolysaccharide. So heteropolysaccharides are formed when different types of the monosaccharide units are joined together. These heteropolysaccharides include compounds like molecules like the hyaluronic acid, which are an important part of the aqueous solution, which actually lubricates the eye, or which even lubricates the joints. The another example of the heteropolysaccharides are the chondroitin sulfate. Chondroitin sulfate are the polysaccharides. Heteropolysaccharides are present in the connective tissue, and they play another very important role there. Thank you very much, students, for giving a patient hearing. Thank you very much.